This video will explore biological buffer systems. Almost every biological process is pH dependent, and even a small change in pH significantly alters the rate of the process. Cells and organisms maintain a specific and constant cytosolic pH, usually near 7, to keep biomolecules in their optimal ionic state. This constancy is achieved by biological buffers. The ionization of water, weak acids, and weak bases is described by equilibrium and acid dissociation constants. The pKa expresses the relative strength of a weak acid or base on a logarithmic scale. The Henderson-Hasselbalch equation relates to the dissociation of weak acids in equilibrium. The formula is pH equals pKa plus the log of the proton acceptor concentration divided by the proton donor concentration. Titration curves reveal the pKa of weak acids and can determine the equivalence point of an acid-base reaction. The pH at the equivalence point depends on the strength of the acid and base used. Buffers are mixtures that resist changes in pH upon the addition of acid or base. Their effectiveness depends on the molar concentration and the relative concentration of the conjugate base and the weak acid. Biological buffer systems are highly efficient. Chemical buffers in the blood act in seconds. The respiratory tract can adjust pH in minutes, and the renal system takes hours to days. Principal buffers include bicarbonate, protein, and phosphate buffers. The bicarbonate buffer system is the principal buffer in blood plasma, consisting of carbonic acid and bicarbonate. It neutralizes stronger acids and bases to maintain plasma pH at 7.4. Acidosis is the accumulation of acids or loss of alkali, causing the bicarbonate to carbonic acid ratio to fall below 20. Alkalosis is the accumulation of alkali or loss of acids, increasing this ratio and raising the pH. The phosphate buffer system acts in the cytoplasm of all cells. It is more effective in the pH range of 5.8 to 7.8, close to the pKa of its components. Its concentration is very low in plasma. The protein buffer system is important in plasma and intracellular fluid. Proteins exist as anions and can accept or donate H+ ions, buffering some carbonic acid in the blood. The hemoglobin buffer system buffers carbon dioxide inside red blood cells. Deoxygenated hemoglobin accepts H+ from the ionization of carbonic acid, buffering it instantaneously. Bicarbonate ions move out of red blood cells and into plasma, causing a chloride shift. In the lungs, oxygenated hemoglobin instantly ionizes, and the released H+ is buffered by bicarbonate, forming carbon dioxide that is exhaled. The amino acid buffer system contains both acidic and basic groups, allowing H+ to pass between them. Amino acid solutions can be acidic or basic, depending on their concentration form.